Hello, ladies and gentlemen. So today we're going to go over gel electrophoresis and how it relates to evolution. So at this point, you guys have all seen pictures like this before, and you know that this is DNA, and these are different bands of DNA. And we can tell if things relate to one another because their bands either match or they don't match. But what we are going to do is look at how these pictures are actually made. Okay, so they are made with a little something called a restriction enzyme. Okay, restriction enzyme. And what a restriction enzyme is it either builds something or breaks down a product. So I put in here, I like to think of it like Bob the Builder. So it either builds something or it takes something down. And in this case, we already learned that cytochrome C is the protein that is formed or that is found in all organisms that have a mitochondria. So what I did down here is I have some abridged cytochrome C sequences from 5 different organisms. So we have humans, gorillas, chimps, old world monkeys, and domestic dogs. So what I did for you guys here is I broke down the bases with 10 at a time, okay? So these are bases 1 through 10, 11 through 20, 21 through 30, and so on and so on. So it's a little bit easier for you guys to see everything. So one thing that we can do, and that we've already done, if you guys remember way back to first quarter, talking about the lions and how we could tell how closely or distantly related they are to each other, we looked at differences. So if we are relating everything back to humans, we can start looking at these differences. So on your worksheet, I have the differences bolded, but I'm going to go through and highlight them here. All right, so I've gone through and I have highlighted all of the differences. So what we can actually do is we can start counting through these things and look at how many differences there are between each of these species and humans. So that's what it is having you do on this next page. So if we're looking at how many differences there are from a human to a gorilla, we can see that the C's stayed the same. We have 1 difference there, so we'll just make tally marks. We have no differences for anything else. So there's only 1 difference between humans and gorillas. Now let's compare humans to chimpanzees. So that one's the same. That one's the same. That one's different, so I'll put a little tally mark there. Oh, that one's different. C went to the T, so another little tally mark. Da-da-da-da. All right. So I'm going to go through and put tally marks for the amount of differences there are. All right. So I've gone through and I've tallied up how many differences there are from humans to each of these organisms. So as we can see, the gorilla has 1 difference. Chimpanzees have 2. Old world monkeys have 4. And dogs have 7 differences. So the organism here that's going to be our out group is going to be dogs. Okay, and a dog is the out group because it has the least in common with everything else. Okay, that's our out group. So what we can do with these differences is we can actually make ourselves a phylogenetic tree or a cladogram. So if we start with the out group, so that's going to be our dog, the one that would branch off first would be the old world monkeys. So those are things like bonobos. The one that would branch off next would be chimpanzees, then gorillas, and humans. So that's how we can start to actually make our cladogram to see who is most or least related to all of these different organisms. So the next thing that we can do with our DNA evidence, because we've kind of already done pictures like that cladogram or phylogenetic tree before, we can now use something called those restriction enzymes. So we're going to make those fancy bands that match up the species to find relationships. The very important part of this is that the enzymes are very specific. So for example, if we had an enzyme program to cut our DNA strand every time it sees the sequence GTTA, where the exclamation point's a cut, our DNA for each of these species would look like this. So down below in these strands, I put a red exclamation point where that thing would be cut. Okay, so in order to make this a little bit clearer, I separately printed off all of these sequences, and I'm going to show you what the restriction enzyme actually does. Okay, so here I have all of the DNA sequences. So we have human, gorilla, chimp, monkey, and dog. What the restriction enzyme actually does is it looks for every single time in the sequence there is a certain combination of letters. So in this case, it's GTTA. So it's going to go along, and whenever it sees a GTTA, it identifies that sequence. And I'm going to do that by just going through and highlighting it. All right, so I've gone through, and if you notice, so this is the DNA sequence for cytochrome C. I very much simplified it. In a real organism, this would probably be like 600 or 700 bases long. So I've obviously made them a lot shorter and simpler. So what happens is we can see between the human and the gorilla, they only have 3 what we call restriction sites. Well, here, the chimpanzee, they had a mutation right there. Okay, and we know not all mutations are bad. So that mutation right there actually made another restriction site. So this chimp's DNA is going to get cut a little bit differently because of that mutation that just naturally happens sometimes. Between the chimp and the monkey, we see that that mutation was conserved, but we also have another mutation, okay? And then between the chimp and the dog, we see that there's a couple more mutations that will add to those restriction sites. So what happens next in the organism is scissors. So remember we said between the T's, that's where everything gets cut. So what those enzymes do when they break stuff apart is they are going to physically cut our DNA into a little pile. Now through movie magic, we have our 5 different piles: human, gorilla, chimp, monkey, and dog, all cut appropriately. So what you get is certain segments. So if we look at the human, we have one segment that is 4 base pairs long. We have one segment that's 15 base pairs long, one that's 20 base pairs long, and one that's 21 base pairs long. So what you can do is go through and figure out how many base pairs long each of these segments are. So now, what do we do with this information? Well, this is where we actually do gel electrophoresis. So what happens is DNA is actually negatively charged, okay? DNA is negatively charged. So what we do is we have a little gel, think like unflavored Jell-O, and then we put this in a water bath and we run electricity through it. So electricity always starts from a negative pole and flows to a positive pole. So when we run electricity through this, we're going from negative to positive. Well, if DNA is negatively charged and we put it at the negative end, so if we fill these little wells with our negatively charged DNA and we run electricity through it, what happens is our DNA is going to start to migrate towards that positive end, and that's what actually creates those cool bands that we see. Little teeny tiny pieces of DNA are going to be able to travel the farthest. So those are the smallest ones that travel the farthest. Big pieces get kind of caught in the Jell-O and they don't get to travel as far. So the big bands are the ones that make bands towards the top. So back to our little pieces, how this actually works is if we start zapping electricity up here, these little pieces are going to migrate down. The little ones are going to go the farthest, and the big ones are going to stay up here. So if we just do this with one well at a time, this little tiny piece is going to migrate all the way down there. We have a bigger piece that's probably going to get caught right about there. We have another bigger piece and a piece that's a little bit bigger. So those are going to make bands that are a little bit closer together. So I'm going to make all of these migrate off camera and then show you what this kind of looks like. All right, so here we can see kind of the migration of all of these things, and we just arrange them according to size. If we have a bunch of bands that are all the same size, so we have here on the dog, we have 3 segments that are 4 base pairs long, that's going to make a really, really thick band on our gel. So what this actually looks like is on the very last page of your assignment. So all of these banding patterns turn into this. So we have all of our different segments up here. They're all mapped out, so that's what you should have counted on the previous page. And then over here, we have our negative charge going in here down to a positive charge. DNA is negatively charged, so it always likes to travel to that opposite charge because opposites attract. And now here, we can actually look at relatedness between these. So here we can clearly see that humans and gorillas, they have all of their bands in common with this one protein. So that does not mean a human is a gorilla. It just means in this one protein, that much of it has been completely conserved. If we look at those two versus the chimp, we have 1, 2, 3 bands that match. So the chimp is decently closely related to the human and the gorilla, but we still have a little bit of differences. So this would be the next most common ancestor. If we look at the chimp versus the old world monkey, we have 1, 2, 3, 4 bands that match and 2 that don't. So a chimp and a monkey might even be more closely related to each other than the chimp and the gorilla. And if you notice here with the monkey, we have 1 band that's thick, so that means that there's multiple sections of DNA that actually have that same amount of base pairs. And then if we look at the dog compared to everything else, we have 1 band that matches here and 1 band that matches here that everything has in common. So we can tell that all of these things have somewhat of a relation. So maybe we can tell that they're all mammals, but the dog definitely has a lot of things that are not in common with the rest of these, like the human and the gorilla, but it still has some things more in common with the chimp and the monkey. Okay, so this is how we can actually start looking at evolutionary relationships. In your lab next class, you guys are going to be looking at this with some DNA from penguins and zoos. So you're actually going to be looking at their DNA and making one of these banding patterns. So I hope you understand how gel electrophoresis actually works a little bit better. If you still have questions, please let me know. Have a great day, guys.